The brain manages its waste through two entirely separate systems. The first operates strictly inside the cell. Through a continuous process called autophagy, a neuron breaks down and digests its own spent proteins and damaged organelles. But whatever a cell cannot digest, or whatever it spills out when it eventually dies, enters the narrow, fluid-filled space between the cells, the extracellular void. Here, the brain faces a mechanical paradox. It is a highly active metabolic organ, constantly generating waste like amyloid-β and tau, yet it lacks the conventional lymphatic vessels that the rest of the body uses to clear debris out of deep tissue. To understand how Alzheimer's disease takes hold, we have to look past the proteins the cell produces and examine how the brain loses its ability to wash away what gets trapped in this interstitial space. For the last decade, the dominant answer to this problem has been the glymphatic hypothesis. This model proposes that the brain solves its plumbing problem by co-opting its own blood vessels, using the tiny spaces surrounding them as a scaffold to flush interstitial waste outward to the meninges. If this model holds, Alzheimer's disease is a sequential failure of this extracellular clearance system, governed by a specific biological switch known as the locus coeruleus. But to analyze this accurately, we have to adopt a strict rule. The glymphatic system is one of the most fiercely debated subjects in modern neuroscience. We must separate the anatomy we know for certain from the fluid dynamics that scientists are still arguing over. We have to grade the mechanics of the machine before we can trace exactly how it breaks. This schematic illustrates the 5 components of the proposed perivascular circuit. It starts with the inflow manifold, the sleeve-like space surrounding penetrating arteries as they dive deep into the brain tissue. The proposed engine driving this fluid inward is the physical pulsation of the heartbeat and the natural rhythmic flexing of the artery wall, pushing cerebrospinal fluid down this perivascular sleeve. To enter the brain tissue, this fluid must pass on microscopic gate. Astrocytes form a barrier around the vessel, studded with highly organized water channels called aquaporin 4, positioned specifically to let the fluid cross into the interstitium. Once inside, the model suggests the fluid moves via convective bulk flow. It acts as a directed current, sweeping dissolved amyloid and tau proteins across the dense tissue towards the veins. The fluid can then collect around these perivenous channels and are flushed out of the skull entirely, draining into the dural meningeal lymphatics, and finally down into the deep nodes of the neck. The entire operation depends on a master switch. During slow-wave sleep, noradrenaline drops. In response, the interstitial space physically expands by roughly 60%, drastically lowering the resistance to flow. When these 5 parts align, the brain becomes a highly synchronized biological washing machine that does its best work only while we sleep. That is a remarkably elegant model, but citing established anatomy to validate contested physics has inflated the field's confidence in how it actually operates. Parts of the circuit are scientifically verified. The perivascular conduits are real. The aquaporin 4 channels exist and degrade in Alzheimer's, and the meningeal lymphatic drains at the top are functionally proven. The heavy dispute is in the middle. The claim that fluid moves through the deep tissue via a fast convective bulk flow is a live controversy. Biophysicists and computational modelers argue that the dense thicket of neurons is simply too resistive to allow a directed current. They suggest transport through this space is dictated by slow, random diffusion, not a rapid, sweeping tide. The foundational claim that clearance increases during sleep is also under active challenge. This 2024 study by Miao and colleagues demonstrated that brain clearance actually reduced during sleep and anesthesia, completely inverting the original rodent hypothesis. Yet, independent of the debated rodent hydrodynamics, human clinical data stands firm. PET scans and cerebrospinal fluid sampling show definitively that a loss of human slow-wave sleep drives up amyloid accumulation. We may not have perfectly resolved the physics of how the fluid moves, but the clinical reality of an extracellular clearance failure, gated by sleep, remains. The biological bridge connecting this sleep-gated clearance to Alzheimer's disease is the locus coeruleus, a tiny nucleus in the brainstem that serves as the sole source of noradrenaline for the entire cortex. This chart tracks its dual role. First, the locus coeruleus is the master engine of arousal. Its firing rate dictates our wakefulness, and it must fall cleanly silent to achieve the slow-wave sleep necessary to open the brain's clearance window. Second, it is the earliest bioenergetic casualty of Alzheimer's. Decades before memory loss begins, hyperphosphorylated tau ignites right here, inside this specific group of cells. This creates a devastating paradox. The locus coeruleus is both the origin of the disease and the switch that operates the clearance system. When it incurs damage, it simultaneously starts the fire and disables the fire suppression system. Alzheimer's doesn't randomly damage brain regions. Its first strike surgically dismantles the exact biological mechanism the brain uses to wash away toxic byproducts. Because of this, the glymphatic collapse happens sequentially. It breaks down across 3 distinct phases of aging. In phase 1, the deficit is purely functional. As early damage to the locus coeruleus erodes our slow-wave sleep, the mechanical plumbing remains perfectly intact. The clearance window simply opens less often and for shorter durations. Over decades, this functional underuse guarantees a slow, subclinical accrual of uncleared amyloid proteins. Phase 2 introduces a structural deficit, aligning directly with the microglial inflammatory phase of the disease. In response to inflammation, astrocytes become highly reactive. As they warp, their precise perivascular gating degrades, scattering the aquaporin 4 channels away from the vessel walls, dismantling the entry point for fluid exchange. By the 7th and 8th decades, phase 3 introduces a hydraulic deficit. Arterial stiffening and arteriosclerosis physically blunt the rhythmic, pulse-driven stroke required to push fluid deep into the tissue. The remaining uncleared amyloid deposits directly into the vessel walls themselves, a condition called cerebral amyloid angiopathy. The exit routes block, the system jams, and waste backs up into the cortex. The disease engineers a feed-forward trap. A failing drainage system leaves behind vascular deposits, which then permanently destroy the drainage routes they sit in. We can now zoom out and view this extracellular failure alongside the intracellular autophagy failure we started with. These 2 systems share a mechanical relationship. What a neuron cannot digest internally is eventually extruded, becoming the extracellular cargo that only the perivascular tide can carry away. When that outward tide fails, amyloid builds up and applies pressure back onto the cell. This rising extracellular load seeds further intracellular failure and aggregation, locking the brain in a self-accelerating loop. In this framework, protein accumulation is the visible residue of a system failing at both stages. Production and clearance are connected stations in a single life cycle. When both tiers of disposal fail, the debris has nowhere else to go. The temporal architecture maps these as sequential failures of a waste management hierarchy, a breakdown that begins in the locus coeruleus and concludes in the vascular graveyard of the vessel walls. Treating a phased collapse requires phased interventions. Our therapies must be matched exactly to the chronological timeline and the established evidence. In phase 1, the most defensible lever we have is protecting slow-wave sleep and preserving locus coeruleus integrity. We can address the functional deficit before the biological machinery physically breaks. Moving into the later phases, the anatomically verified meningeal lymphatic drains offer a viable preclinical target. Supporting this efflux route using vascular growth factors acts on a structurally sound part of the clearance model. Systemically, managing hypertension and arterial stiffening serves as a direct intervention. Preserving vascular health maintains the mechanical pump required for periarterial influx. While biophysicists continue to debate the precise hydrodynamics of the interstitial tide, the evidence points toward a progressive failure of the sleeping brain to wash itself. Delaying that failure means protecting the switch, long before the pipes have a chance to clog.